New day, new vlog, good morning. So today is going to be a very good day because today we have a seminar here at the UCC and the title of the seminar is Pathogen Without Borders. And the good thing is I'm going to do a presentation as well. So although I'm here for just 1 month, I felt like it's very good to present about knowledge that I know and share with the people. So the audience are going to be public. They are not, all of the audience are not scientists. So the presentation will be very basic in a level that public will be able to understand. And now I'll head towards the hall where we are going to do the seminar. And the good thing is that the seminar is going to happen inside this building. So let's go. So the seminar is happening upstairs. I can see that it's here, the poster. So we are going upstairs. Seems like it is in this room. Yes, it is here.
We'll start. Good morning, everybody. This symposium, we have been able to run this thanks to funding from a Research Ireland call, which is between Ireland and Wales. So it's the Wales Innovation Network. And we have our Welsh partners are here. And I suppose, firstly, thanks to the two Johns, John Doonan and John Mackrill, who are PIs on this funding, and Lewis and myself would be the collaborators. We have asked them to sit in the King's chair at the bottom because they're the PI, but they're refusing to do that, but anyway, that's okay. Okay, so the idea of the funding is to bring people together like we've done today, with a view to putting in a grant application to Horizon Europe, which we're also doing. And it's one of the reasons we're having the meeting, is to get people together to share ideas. And of course, we want to, and we have opened this up to the public. And I suppose maybe the time of year that it is means that we have a few enough members, but we're delighted to see members from outside our grouping as well here. So and it's to facilitate, good morning, to facilitate wider discussions between Ireland and Wales, and we're confident that's going to happen. Okay, you all have the agenda. There's a printed copy of the agenda here if you'd like it. And so we have speakers from UCC, quite a few. We have speakers from Aberystwyth in Wales. We have a speaker from Trinity. We have Tyndall National Institute represented. We have Teagasc, and we have the Norwegian Research Centre as well, the Department of Ag. Did I mention Department of Ag as well? So, you'll hear a lot about maybe Phytophthora infestans, but there's lots of different species of Phytophthora. And at the latest count, I think it's put at about 326 species. Many of those identified in the last 2 decades, actually, thanks to kind of improved molecular techniques, etc. So about myself, so I'm Barbara Doyle Prestwich, and I'm just going to kind of set the scene and then we'll move on to different speakers. So I'm by trade a plant biotechnologist, and over the years I've worked on a lot of different crops, but I suppose what brings them all together is that all of these crops are vulnerable to disease, and we have to have strategies as a way to deal with this, otherwise we lose our crops. So Phytophthora infestans, late blight, we're all well aware from our history books of the problems that this has caused, especially on the island of Ireland. So the Great Irish Famine in the middle of the 19th century, where over 2 million people, so there was devastation on the island of Ireland in part because of the devastation of the potato crop, because of Phytophthora infestans at that time. So it was a staple, and when infestans came, there were real problems for people. Actually, the plants that you saw circulating there in the tub is the variety that was grown at the time in Ireland called lumper. So we keep lumper, we don't really work on it, but we keep it, would the right word be nostalgia? I don't know, but from a historical perspective anyway, we keep lumper just for these occasions, okay? Obviously the famine had a big social impact on Ireland at the time, but of course there was famine across Europe. But in Ireland, we dealt with it maybe slightly differently, or didn't deal with it. The population today in Ireland is still below pre-famine times. Infestans is still a problem globally, so that hasn't gone away. In Ireland, farmers spray up to 15 times per season for this particular, just one disease. The global production of potato you can see there, plus the loss. But look, it costs about 10 million. So it's a real economic problem to control these diseases as well. It costs over 10 billion to deal with one particular disease, which is phenomenal. It does depend on where in the world you live. If you can afford to spray, you spray, because it's chemicals really is controlling plant disease. That notwithstanding, under kind of directives from Europe, we are asked to reduce our pesticides, the number of pesticides that are available. And this is causing problems for growers. If you look at different parts of the world, like Kenya and Uganda, for instance, where there are over 1 million smallholder farmers, they suffer losses of up to 70% because of disease. So it's a huge problem, okay? But chemicals in the main. There are other ways, and you'll hear more about other ways today as well. So we have something called integrated pest management. I don't know if members of the public, I'm sure you might have heard of integrated pest management. And it just means that a whole plethora of measures are used to control disease. Crop rotation, for instance. Use resistant varieties where possible, but it's not always possible. So in organic farming, and again, the organic farming community were invited today, but maybe it's that time of year as well. But we will engage with them for the Horizon Europe project as well. In organic farming, certain varieties are used that have a greater level of resistance to Phytophthora, but those varieties are not as commercially popular as some of the ones that we would be familiar with, like Cur Pinks, Golden Wonders, etc. Do you all eat potato? Yes. So we can see how valuable this crop is to all of us. How an evolutionary biologist, a physiologist, and a cancer researcher is flying the flag for Phytophthora. We're going to hear how tools being developed at the lab bench, how they will ultimately help in our quest to control Phytophthora. We're going to hear about sudden oak death. So it's not just Phytophthora infestans. As I say, we have multiple species to deal with. We'll hear about previous projects on infestans, and we'll hear about the National Plant Phenomics Centre in Wales. We'll also hear about multiomic assessments of plant-fungal interactions. We'll hear how those same microbes that I spoke about earlier, where and how they are sourced in order to control infestans. We'll hear about the plant health in Irish nurseries. So what is the disease like in Irish nurseries and how we're keeping track on that. We'll hear about artificial intelligence and hyperspectral imaging and how that relates to monitoring disease and health status of plants. We'll hear about, again, plant nursery pathogens and using high throughput sequencing technology. And again, we're going to keep it in a way that we can all lean into the talks, and understand. And then the final talk is from our Norwegian researcher, Bikal, who will talk about broad omic technologies. Okay, so some of the terms, if you're a general member of the audience, don't panic. Because we're going to keep it in a way that's very accessible to everybody.
And my projects are on two main areas. One is potato blight and the other is on esophageal cancer. So two very different areas, but they are connected, and I'll hopefully get that across. So what is potato blight? Barbara's already explained this very well, so I'll keep this very brief. Phytophthora infestans. So if you look at the name, scientists like to use complicated names for things, Latin names. Phytophthora is the genus. So we're Homo sapiens, but there are other members of Homo, like Homo erectus, Homo neanderthalis. So they're all members of the same genus. Infestans is a specific species equivalent to our sapiens. And they're oomycetes. So oomycetes are fungus-like organisms that, they resemble fungus, and they're also commonly called water mold, so a family of different organisms. They look and act like fungi, so this is, Andrew, who's an expert, is going to show you more of these, but this is how we culture them in the lab on this little plate here, and this is what they look like under a microscope. You can see that they're 24 millimetre in width, so very, very small microorganisms. So human hair is much bigger. And in terms of what they actually are, even though they look and behave like fungi, they're actually closer to algae than they are to fungi. If you see this little family tree, we're closer to fungi than potato blight are. They just resemble potato blight in terms of what they do. So how do you control late blight? One way is prevention, so monitoring. So you're looking to see when the blight appears. So looking at the weather, looking for the appearance of disease. Another way is to remove coal piles, so piles of waste potatoes and volunteer plants. So these are things that sprout from the waste basically. so remove and destroy them. And another one is to grow, as Barbara said, resistant varieties. These can be either naturally occurring or genetically engineered, so there's big resistance to genetic engineering. Another way is crop management. The application of fungicides is the key method. Expensive and potentially harmful to the environment, there's a limitation there. And there are also newer organic, ecological, like the microbiome that Barbara was talking about, biological approaches. And there's also cure, destroy the infected plants. So, Phytophthora infestans, this is the key point I'm trying to get across the Bristol, is only one species of over 1,200 different species of Oomycetes. So some of these cause infections in most species of plant. I think every species of plant I can think of really. And they cause different diseases like downy mildews, root rots, seed rots, and damping off of seeds.
Okay, so good morning everybody, and welcome. My name is Andrew Foley. I am an MSc research student who has had the pleasure of being part of the Oomycete research group here at UCC. I work closely with both John and Barbara, and I suppose it pains me to say it, but I'm going to the end of my 2-year research project where I've mainly been looking at, I suppose, protein biology and some of the calcium signaling mechanisms that John mentioned, in particularly Phytophthora infestans. But my goal for today is to speak very generally about some of the techniques that I use and people like me use to study Phytophthora species, I suppose, from a physiological and molecular biology angle. So, I'm not going to dwell on this too long because I think John has covered a lot of it, but I think this slide highlights really well, I suppose, the complexity of the life cycle of Phytophthora infestans and Phytophthora species in general. It's multi-stage, it's very dynamic, and it's very diverse. So I think it's important as researchers that we also employ an equal degree of diversity in our methodology. So today, I'm going to cover some of the topics like culturing and maintaining Phytophthora in the lab, and that's what you can see up here in the top left. From the angle here, I don't know if I'm hitting it, but somewhere up there. And I'm going to talk about using microscopy, various techniques to visualize Phytophthora structures up close, talk about some controlled infection experiments, and then go a little bit closer to, I suppose, the work that I've been doing over the last few years and talk about DNA and gene expression, and then protein biology and cell signaling as well. So, beginning with culturing, we culture Phytophthora on agar plates, which are essentially jelly-like plates. I'm going to hand these to Barbara to pass around. They are completely clean and sterile, no Phytophthora, so if you want to open them up and have a poke around, go for it by all means. But we supplement these agar plates with extract from rice seeds, and then we mix that with sucrose, sugar, and then that acts as a very efficient nutrient medium for the Phytophthora to grow. And it kind of grows in a fluffy, I suppose, almost cotton wool-like manner, out really from the centre where we inoculate it. And then we store those 18 to 20 degrees in the dark, and that allows us to culture hyphae, sporangia, or even induce the production of zoospores. And culturing and maintaining Phytophthora species in this manner is very powerful in itself, as it allows us to do things like growth assays, where we can supplement these rye agar plates with potentially new anti-Oomycete treatments, and then monitor the effect on growth over time. So as you can see, as time goes on, we can see, I suppose, the cotton wool-like structure emanating from the center.
I'm going to tell you a bit about Phytophthora amorum, and I just want to say thanks to Barbara for inviting me. These are new results. I've been excited for a couple of weeks to tell somebody about this because we got these. I've been waiting for these results for about 10 years. We started a postdoc project in 2012, and we did a lot of morphology work on Phytophthora amorum, but the one thing I was always missing was any kind of genetic analysis of population studies. So you're the first people to hear about this, so I'm delighted to be able to share it. So Phytophthora amorum is mainly, well, when it was first described, it was mainly a pathogen of horticultural plants. These are rhododendron. The one on the left is heavily infected, the one on the right is healthy. So had a severe effect on hardy nursery stock. This was when it was first found. There was a problem in nurseries, but as these things happen, it then spread out into environmental areas, parks, forests, etc., and started causing a lot of disease in North America as well as in Europe. It's got a huge host range, I think over 200 or 300 different horticultural nursery plants. It was found in Ireland first in 2002-2003 on rhododendron and viburnum. And then the Department of Agriculture, where I work, we started monitoring for this because it was a quarantine pathogen. It was regulated. You had to destroy it. We started monitoring in nurseries, and we were catching up on it. We were taking lots of samples, finding lots of positives, leading to lots of action. Then in 2010, the game changed a little bit. It was found in forests.
Right. Well, I'm John Doonan. Thank you very much, you guys, for organising this and having us over to Cork. So lovely weather.
We arranged that.
Anyway, so I'm from Aberystwyth. I'm part of the UCC funded programme in resilient crops. Essentially, what we work on, a variety of crops, mainly to do with western part of the UK and Ireland, forages, potential biomass crops, resilient cereals like oats. And we look at the pre-breeding, pre-commercial breeding part of this, and a lot of the stuff that is produced within the department goes on to these linked seed companies, Germinal, Sonova and Paravesta, for commercialisation. So there's a number of programs in the actual breeding of those within the companies and also within the department. But the particular thing I'm going to talk about today is something called high throughput phenomic technologies and how that might be applied to breeding. I should also point out that part of the breeding process is very much focused on disease tolerance. So one of the selections that all of these crops go through is they grow them up under really shitty conditions that allow a variety of diseases to take hold, and anything that survives that goes on to the next stage for other traits. If it's not resistant to disease, there's no point.
So I'm Lewis Smur, okay, from Aberystwyth. People know, we're kind of introducing ourselves, so actually I've got a North English accent, but my parents both Spanish, so that's why I've got a kind of non-traditional UK name, so it's Smur actually, that's why I've got recent hands with it. So basically, nice to be here, and I'm going to be talking about plant work, and plant-pathogen work, and particularly looking at this thing called metabolomics. One of the workers that Andrew basically referred to, that is the output of the genome sequence variation. So I'm highlighting the capability that we've got to actually hopefully collaborate with you guys in a lot of detail, okay? Before I go into this work, I'll mention that rather like John M, I actually have a lot of human work underway, human clinical work, and also veterinary clinical work. We even actually build lateral flow systems, and we have a lateral flow system under testing in Ireland for the detection of bovine tuberculosis, and we're doing that with University College Doyle. Okay, so we're talking a lot about our work on plant pathogens, but we have got a lot of interest, a lot of expertise, which we're happy to share and explore with you as relevant. So this is kind of like very similar to I apologise first, it's a bit more scientific, okay, but I'll actually skip through things and say what's the take-home message, because I know there's a very varied audience in front of me. Okay, so here is basically the bosses, the genomics. Okay, we've got a lot of workers, that is the proteins, or proteomics, the genes, called transcripts, hence transcriptomics, or the gene outputs, which are the RNA transcripts, and metabolomics. Okay, so metabolomics, basically we use it for establishing what we call a metabolic or biochemical phenotype. We look at novel targets for breeding, for resistance, or even simply understanding what is going on in any particular interaction. And we also look at biomarkers. And hence, the last thing here basically is kind of like associated with plants, but also more often associated with the clinical context. Hence, you can see why I'd say probably 60% of my work actually is the clinical and veterinary sphere.
My research expertise is in the use of soil biodiversity, either to study how they interact with plants or how plants interact with them. So in this work that I did when I was a research fellow in University College Dublin, I'm going to talk to you about the role of endophytes to control potato leaf blight. So endophytes, what are they? They are the group of organisms, often mostly bacteria and fungi, that live inside of plants. Just like humans have got guts, plants also have their guts. Mostly they wear it on the outside, but these ones live inside the plants. And the entire community of organisms that are associated with plants is what we will call the plant microbiome. So these organisms can actually help plants to tolerate biotic and abiotic stress. So we set out with this project to look at how we can learn from nature to nurture, to learn from what is happening in the wild, to see how we can bring that into food production. So we started off with looking at diseases of cereals, particularly Fusarium graminarium of wheat. Then we branched off to look at horticultural crops. So we're looking at lettuce, we're looking at strawberries, we're looking at potato.
Hi everyone, I'm Kes. I'm a PhD student on the Dept PMP project with Michelle and Richard here. I'll be talking to you today about some eDNA insights into disease presence and plant health in Irish nurseries. So I'm covering a bit of the ground that Richard covered earlier about how much of a risk nurseries are for pathogen spread. The import of infected plant material into nurseries is believed to be responsible for about 69% of pathogen outbreaks worldwide, and it's the main pathway of spread for oomycetes internationally. So there's large numbers of plants being grown in really small areas, so disease detection is quite challenging, particularly because a lot of infections are asymptomatic, and often it can loads of different plants growing in close proximity can facilitate the spread of pathogens from asymptomatic carrier plants into other hosts which may not have the same immunity. The Agri-biotech project in the UK found 85 Phytophthora species or complexes in 134 British nurseries. And then there's a few photos just showing the damage that those species can cause in the wild then if they escape. And one example of which Richard spoke about earlier as well is I just have a timeline of Ramorum in Ireland. So it was detected in garden centres on imported plants in 2002. It was detected in the wild in 2003. And then by 2010, it was in Japanese large forests, and it's a serious ongoing threat to forestry now. And there's some photos just showing the level of damage it causes in Japanese large forests in Ireland there.
So, hello everyone. My name is Matteo Melotto. I'm a senior researcher at Tyndall National Institute, which is the leading national institute in ICT and photonics, with a whole cascade of applications, of course, that goes from quantum computing to the development of new imaging sensors, biochemical sensors, and so on. So I've been tasked to give you an overview of the new trends, let's say, on hyperspectral imaging, and especially in combination with AI. So imaging, let's say, to walk through a field of crops, everything looks green and healthy, but beneath that surface, something is changing, stress is building up. It could be nutrients deficiencies, it can be early stages of a disease, it can be pest activity. The problem usually is that when that is visible is often too late. Modern or current, let's say, ways to detect these kind of conditions are mostly rely on visible signs, and of course these could be an issue. And here comes the promise, in a way, of hyperspectral imaging, which can allow to see what is invisible at naked eye. So when we combine hyperspectral imaging with AI, it doesn't just see, so to speak, but it understands. And I will explain a bit more that concept later on. This presentation will give you just an overview of what these two power tools combined together can actually promise in the field of agriculture in terms of precision management or sustainable growth. To understand hyperspectral imaging, think of it as a cameras, let's say, with superpowers. Our eyes and also normal cameras can see, roughly speaking, in three colors, red, green, and blue. But in hyperspectral imaging capture hundreds of spectral bands. And each pixel in an image contains a full spectrum, effectively. And as you know, probably more than me, and that spectrum is basically the fingerprint, in a way, of a material. So we know a lot of things just from even a single pixel. So this means that we can detect things like chlorophyll content, water stress, fungal infection, long before their manifestation is visible in the leaf or in the plant in general. Why does this matter for agriculture in a way? So because plants respond to stress in ways that change their reflectance, which is ultimately what we capture with hyperspectral images, especially in the near infrared and in the red edge. Hyperspectral images can pick up these changes very early. For example, again, nitrogen deficiency is a classic which alters the leaf reflectance, but also fungal infections is another classical things that we can detect with hyperspectral imaging, as it shifts the water content and the pigment levels, which are detectable spectrally speaking way before the lesions appears. Hyperspectral imaging generates a huge amount of data, and a single image can contain even millions of spectral signatures, which of course it's an issue in terms of data analytics. And here is where AI comes in hand, in a way. Deep learning models like convolutional neural networks and others, which we will see in a second, are very, very good in detecting patterns in the spectrals. And so we can use these techniques to effectively classify plant health and also predict disease basically on set, or even recognizing hyperspectral data from classic RGB images.
Hi everyone, I'm Michelle Thompson. I'm from Chagas, and I'll be talking about the Detect PNP glasshouse experiments. So this is in connection with Richard and Tessa's presentations earlier. So I'll be talking about detecting plant nursery pathogens using environmental DNA and high-throughput sequencing technologies. Didn't cover it earlier, but we know that invasive plant pathogens are a problem. They're a danger to the environment and the economy, and some Phytophthora have wide host ranges, so they can jump from plants that are imported into wild plants. In our experiments, we don't just want to know if one plant is sick, we want to know if we can detect different numbers of infected plants within a healthy group. So if we're looking at 5 green healthy plants here and 1 infected plant, we want to know how different that might be from testing water that comes from 3 healthy plants and 3 uninfected plants. So we'll be adding to culture collections at FM and making this available to researchers in Ireland. This is important because good high-throughput sequencing requires good reference databases. So we need to know what we're looking for. And the more we know about the diversity of UCC in Ireland, the better we're able to identify them.
Yeah, hi everyone. It has been a very wonderful day with lots of knowledge. And I will try to top it off with some cake, as I'm the last speaker, I suppose. I come from Norway. My name is Bikal, and I'm a researcher at Norwegian Research Center in the group Gene Technology, Environment and Society. And we are also the competence center for biosafety and biorisk assessment in Norway. And I'm very happy to be here to present some of the knowledge, and thanks to Barbara for this wonderful opportunity. And in today's talk, I'm going to provide some brief information at a very basic level how we researchers use the information from plant and pathogens to develop strategies to control disease in the field. That's why the title of my presentation is Translating Pathogen and Plant-omics to Control Crop Disease. And let me remind you that by the year 2050, the population of the world is expected to be around 10 billion. And to meet the food demand of that population, we have to increase the production by 60% than that of today. That was stated in the International Year of Plant Health 2020. So, protecting plants is very crucial to protect life. And we have come across a lot of devastating disease caused by pathogens in the field that have led to great effect, devastating effect like Irish potato famine that we all are aware of. Plant pathogens like fungi, oomycetes, virus, bacteria causes devastating crop disease. That's why plant disease is a serious threat to global agriculture. And as today's theme is based around potato and leaf blight disease caused by Phytophthora infestans, I'll try to give you an overview how Phytophthora species cause disease in the field.
And the omics approaches, you can see how powerful those are. But I think the world's changing rapidly and we are trying to keep pace with it, really. It would be my opinion. That's just my opinion.
Yeah, I agree. I think we describe it as an arms race. Actually, trying the pathogen will evolve. You know, new strategies. There are a number of different strategies that can be used. Some are more acceptable than others, and some are slower than others.
Is that there are in Ireland areas of historical copper mining and other areas with anomalies with copper. And I was wondering, and there's a lot of very good data with all these minerals in the soil in Ireland. Have you noticed a difference in resistance in copper mining areas to other areas?
I'd say it's a good question. This is the sort of thing that we want to hear, you know, I think, because I hadn't thought about that at all, but you're absolutely right. There could be a difference in kind of prevalence even of the disease, you know, because of local geographic conditions or geological conditions. But
Things before they could. How often do you have to do that to be, you know, in one season to be effective, or do you just do it once, or?
Literally taking a picture of a plant, so it depends on at what stage you take the picture, you get a response in terms of presence or no presence of pathogens, disease or pests and so on. So it's, you know, it's easy enough, I would say, to take a picture, especially if you have a drone.
So yeah, that is it. It has been a great day with lots of learnings and knowledge. Different researchers sharing their insights about how we use the plant and pathogen information to control disease in the field. It was a great day, and I hope you enjoyed the vlog and helped share the knowledge to other students and audiences. And I will also post individual presentation, full individual presentation in the playlist that I will create, and I hope you enjoy watching those presentations and also help by sharing those presentations so the public can be aware about how we use science to control crop disease in the field. So yeah, thanks for watching, and I hope you subscribe my YouTube channel for more updates. Thank you. Bye bye.